Hello everyone and welcome to today's presentation, Rapid Hyperplex Staining and Simultaneous Imaging for Immunophenotyping of Tissue Sections. I'm Irene Tamayo, Head of Marketing and Communications at Lunaphore, and I'll be your moderator for today's event. Today's webinar is brought to you by LabRoots and sponsored by Lunaphore. So if you would like to have any more information about us, Lunaphore, please visit our site at lunaphore.com. Before we begin, I'd like to remind everyone that this is an interactive event, so we encourage you to participate by submitting as many questions as you want at any time you want during this presentation. And to do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located at the far left of your screen. So now I'd like to welcome today's speaker, Dr. Diego Gabriel Dupouy, Chief Technology Officer at Lunaphore Technologies. Today, Dr. Dupouy will introduce you to our microfluidic technology and our product Comet, currently in development, that features integrated imaging and staining of tissue samples. He will then show you how the staining and imaging integration is achieved, and also how we optimize assays to obtain hyper-plex results in short turnaround times, thanks to our novel microfluidic technology. And finally, he will focus on protocol transfer to different tissue types, and after which we will have a live Q&A where we, as mentioned before, we will answer the questions received during the presentation. If you would like to know more about our presenter, please visit the Biography tab at the top of your screen. So with this said, I think Dr. Dupouy, you may now begin your presentation.
Thanks, Irene, for the nice introduction. Good morning, good afternoon, good evening, everyone. It's a great pleasure for me to present at this LabRoot webinar. Our product under development, Comet, is an all-in-one staining and imaging platform that integrates our microfluidic-based staining with a fluorescence microscopy. It can run extremely rapid hyper-plex assays in an automated fashion. Since it's an open system, it allows the use of label-free, off-the-shelf or custom primary antibodies. For more information about our product under development, do not hesitate to reach out to communications@lunaphore.com. The core of our technology is based on a microfluidic chip called MTP for Microfluidic Tissue Processor. It is used to reach high-performance automation of in situ staining techniques on tissue samples, such as high-plex immunofluorescence. The chip is a set of microfluidic channels designed to deliver and wash reagents on the vicinity of the sample. It is clamped over standard histological glass slides, forming a closed chamber of reaction over the tissue. We call this technology FFX for fast fluidic exchange. This name is due to the fact that pressure-driven reagents flow into the chamber and the liquid exchange is very rapid. The perfect sealing offers a controlled environment with multiple benefits. Staining times are reduced due to the active transport of reagents, a more uniform staining over the tissue surface is achieved, and evaporation is avoided. A heating element placed below the slide allows for precise temperature control and at any time, as well as rapid temperature changes thanks to the limited heat capacity of the assembly. Finally, our microfluidic chips have an integrated glass imaging window that allows for integrated staining and imaging. This way, the user does not need to move samples from staining stations to imaging stations, neither perform multiple cover slipping steps. Our technology has been widely validated across a number of studies and publications. We've demonstrated that running microfluidic-based immunofluorescence assays with short incubation times, we can better quantify the fluorescence signal from the tissue thanks to the avoidance of saturation. We could even classify ambiguous HER2 status of breast carcinomas into FISH positive and negative, basing ourselves on the quantification of the fluorescence HER2 staining. We have also shown that we can obtain extremely rapid chromogenic and fluorescence stainings on different markers of both formalin-fixed paraffin embedding and frozen samples. Additionally, we've shown that DNA and RNA can be preserved during the staining performed with our instrument, and the samples can be further utilized after staining for next-generation sequencing. With our technology, we can also run fish assays and target RNA expression. You might have watched our seminar showing the capability of our product LabSat to run pyramid signal amplification-based multiplexing assays in combination with the Opal kits from Akoya. If you missed it, you can find it in our website or just get in touch via email. Finally, in this presentation, I'd like to introduce you to the use of our technology to perform multiplexing assays in a sequential fashion. Our sequential immunofluorescence assay consists of the following steps. We incubate a cocktail of 2 primary antibodies of different species. Subsequently, we incubate a cocktail of 2 fluorescently labeled secondary antibodies. And we acquire the image. We then remove all the antibodies using an elution buffer and heat. We repeat this process over and over again. The instrument will automatically register, stack, and stitch all the images. Every sequential immunofluorescence cycle consists of the incubation of 2 primary antibodies from different species. In our case, mouse and rabbit, for 2 to 4 minutes, followed by a 2-minute incubation of a cocktail of 2 fluorescently labeled goat antibodies, and finally a 1-minute incubation with Dupouy. The overall staining cycle lasts 10 to 12 minutes. After the staining is finalized, the tissue is scanned through the imaging window of our microfluidic chip. No decoupling between the staining chip and the sample is required, and the process is performed in an automated fashion. In the data that I am showing today, we scanned areas of 4 by 4 square millimeters. The total cycle of staining, scanning, and elution takes around 25 minutes, generating 3 stitched images, Dupouy, and 2 markers of interest. We can repeat this cycle for as long as we'd like to. We have shown results for up to 15 cycles, giving 30-plex staining as of today, but we expect this count to keep growing. Once all the images have been acquired, the registration and stacking process takes place. Before a multiplexing assay can be performed, we need to go through a series of steps of optimization. Our technology allows for playing with multiple parameters at temperature, incubation time, the use of dynamic incubations to achieve more homogeneous stainings, perform multiple flashes, etc. To achieve a good multiplexing experiment, we make sure that we can remove the signal of every staining in order to avoid non-specific signal appearing downstream in the protocol. For this reason, every marker goes through an elution characterization step, where we make sure that at least 99% of the signal is removed. We compare the remaining fluorescence after elution to the autofluorescence of the tissue obtained prior to starting the optimization procedure. A similar background level indicates an efficient elution of secondary antibodies from the tissue. Subsequently, we perform a controlled staining using only the secondary antibodies and compare the fluorescence signal with a reference negative control. The scope of this step is to highlight any remaining primary antibody after the elution. Once marker optimization is achieved and the elution efficiency is proven to be above the acceptance criteria, every marker goes through an epitope stability characterization step, where multiple cycles of elution are performed, intercalating staining controls in the process. In the example of this study, we show an initial staining of CD20 followed by 10 cycles of elution and the second staining. After 15 elution cycles, a third CD20 staining was performed. We compare the signal intensities and background levels for each step. The The information gathered during elution efficiency and epitope stability characterization is fundamental when creating our multiplexing panels, as it will determine how difficult it will be to remove the signal from a particular marker and how stable the epitope is to multiple cycles of elution. This ultimately guides us on what position to assign each marker to in our multiplexing panel. Once every marker in our panel has been characterized, we are ready to create a multiplexing protocol. In the example you see in this slide, 24 immune markers were stained on formalin-fixed paraffin-embedded human tonsils. The scanning area was set to 4 by 4 square millimeters, and the total time of the experiment was 5 hours. We also use chromogenic references for every marker as the benchmark for the areas we were expecting to see signal. Subsequently, we transferred our protocols to samples of non-small cell lung carcinomas and compared again with the chromogenic gold standard. We know that performing quantitative comparisons between chromogenic and fluorescence images is not straightforward, so we came up with two simple image processing algorithms that would allow us to determine the area of the tissue that was stained both in chromogenic and fluorescence and plotted those values in a scatter plot, both for tonsil and lung carcinomas. We could appreciate a very good correlation between both stainings in both tissue types. It is widely accepted to employ tonsil samples during the early optimization of immune markers due to the easy access to tissue and the abundance of the expression of such markers. However, this comes with additional difficulties. Very often, we need to perform some re-optimization steps once we transfer to specific tissue types. In some cases, only one or a few tissue types require adaptation, while in other cases, a more general re-optimization is required. For example, this was our approach to transfer this 7-marker panel from tonsil tissue to colorectal, cancer, lung carcinoma, and melanoma. When we attempted to transfer CD3 and CD4, screening confirmed no need to further optimize on lung and melanoma. We increased primary antibody and secondary antibody concentration in colorectal tissue for optimal results. We would typically look at readjusting the concentration of primary antibodies first and/or the incubation time. As a second step, we could also play with the concentration of the secondary antibody and decide or not to include an additional blocking step. For FOXP3, PD1, and PDL1, we required increasing only the secondary antibody concentration on colorectal and lung tissue. For CD68, we increased primary antibody concentration on all 3 tissue types. And finally, in more difficult cases, even if a starting tonsil protocol seems to work, the transfer to tissue would require the choice of an alternative clone. This was our case for CD8. To conclude, tonsils helped set the starting point for many markers during the optimization of our multiplexing protocol. However, when transferring to specific tissue types, a subgroup of the markers require further adjustment. Our technology allows for testing of multiple conditions in series with limited need of transferring the samples from a stainer to a scanner, avoiding many manual steps of cover slipping and decover slipping. I'd like to show you a sample image of a transfer from tonsil to colorectal cancer that we obtained in collaboration with Professor Inti Slovec from the University of Bern in Switzerland. We have optimized a 6-plex panel on colorectal cancer as the preliminary steps towards a larger panel. In this case, we applied a background removal algorithm that was developed in-house and that takes into account the autofluorescence of the tissue acquired automatically at the beginning of the assay. The last step of this presentation is simply to highlight that the images generated with the COMET instrument are exported in OME-TIFF format, making it possible to import from most tissue analysis tools. On the left-hand side, we perform cell segmentation using the STARTIST algorithm on QPath. Then we train a classifier to recognize FOXP3 positive cells. On the right-hand side, we used an AI-based algorithm from Halo for cell segmentation on one of our COMET images. I hope I have been able to show how the use of microfluidics allows for rapid and efficient staining and elution cycles of tissue samples. We've discussed how our technology can be used in combination with an integrated fluorescence scanner and how this enables full automation. And finally, it is important to mention that the product we are developing combines an integrated staining and imaging workflow with 4 slide capacity. COMET will allow researchers to run their project without compromising in terms of plex level or number of samples. My team and I are now happy to answer your questions.
Thank you very much for your presentation, Diego. Before we start the Q&A, I want to mention that we will be sharing this recording on our company website, so www.lunaphore.com. So let's start with the Q&A. We will go through as many questions as we have time for, but if we're not able to answer all of them, we will surely get in touch with you to reply any pending questions. Or if you would like to submit any more questions after the Q&A is over, you can also do so via email by sending an email to communications@lunaphore.com. All right, so let's go ahead and get started. We have already some questions coming in. The first question that we have is, can we use any desirable marker? Is the platform open? Any requisites for antibodies?
That's a very interesting question. So yes, the platform is open, so we can use any marker of interest. There is no particular requirement, neither from the point of view of barcoding or whatsoever. The only specification that we need is that the fluorophores that we use in conjugation with the secondary antibodies have the right match in terms of emission and excitation spectrum.
Great. We have another question here regarding the type of sample. So this question is asking, what is the thickness of the sections that we can use?
So we typically use samples of FFP samples mounted on standard histological glass slides of about 2 to 5 microns thick.
Excellent. Another attendee was asking about if you could clarify how you erase the signal in this application.
Yeah, this is a very important point. We've been optimizing on this since the very beginning of the development. So it's a combination of low pH and a detergent and heat. And what we do is we elute, so we really remove the complex of primary and secondary antibodies from the tissue.
Excellent. I see more questions coming in. This is great. So please go ahead. We have another question asking, can we identify intracellular markers?
Yes, we have experience. I mean, we have data showing that we can detect all types of markers, membranous markers, cytoplasmatic markers, and nuclear markers. We have, for example, optimized FOXP3, Ki67, etc.
Great. Someone else is asking, is it possible to use cocktails of antibodies in a single staining and imaging cycle?
Yeah, this is a very important question. As I showed in the presentation, so basically we designed the experiment so that we can target 2 markers per cycle of off-the-shelf antibodies, normally of 2 species, typically 1 mouse, 1 rabbit. So yes, we can cocktail, but they don't necessarily need to be mouse and rabbit.
Great. Another question on sample type. So this question is asking, is this method restricted for use on specific tissue types, or it can be used on any tissue type?
So we have experience with different tissue types. both normal tissue and cancerous tissues. So there is no restriction from that. And on top of this, we are very interested in frozen sections as well. We have plans of developing the staining on frozen sections, and we already have experience on the staining itself. I'm sorry, we are developing now the multiplexing protocol for them as well.
Excellent. Another question here is asking whether antigen retrieval is performed prior to this application or automated on the device.
This is very important. So the antigen retrieval needs to be done offline, so on another instrument prior to be mounted into loaded into our system. But we are optimizing it with a module that is automated. However, the user can choose what method to use for the waxing and antigen retrieval. The way we're suggesting is using an instrument in order to get it automated.
What about the maximum sample type size that you can evaluate?
So the evaluation area is a maximum of 9 by 9 square millimeters. And we are interested in going further in the future, but this is our current area that we can evaluate.
Excellent. We have another question coming in. Someone is asking what resolution or zoom can you use or can you achieve with this device? Are details visible at the cellular level?
Right. So we are going to reach under the 0.5 micrometers in resolution. We're going to do so by having a rather high numerical aperture objective and in combination with a very high quality CMOS camera. As I mentioned before, we can target markers that are either nuclear, cytoplasmatic, or membrane. I don't think that with immunofluorescence we could go any further than that in general. Let's say an optics limitation.
Another question coming in. This one is saying, could you please give a rough estimation of the staining time per sample for a 20-plex panel?
Yes, so we do have an average, but it's a little bit vague, so we'd rather give some examples of it. So for like, I think one of them I showed in the presentation, a 24 marker panel scanning 4 by 4 square millimeters takes 5 hours. And another example of 30-plex took us 8 hours for a 9 by 9 square millimeter area.
Excellent. Someone else is asking here, what's the maximum number of markers? I think it says tried with this technology. I'm guessing that's the typo. Tried with this technology.
Yes, we have data so far showing 30-plex with our technology, but the count keeps increasing as we speak.
What is the actual estimate of number of plex that we will deliver with COMET? Maybe that is also an interesting question for our attendees.
So we designed the instrument to reach 40-plex, but we are not setting ourselves an upper limit for it.
Some attendees are asking about some more details about the microscope. So what can you tell us about the microscope coupled with COMET? So for instance, magnifications, can we choose one area or more, etc.?
All right, so the COMET has one objective. So in terms of objective magnification, it's a 20x. However, because we use a rather high numerical aperture objective, we can zoom in and look at the tissue as if it was a 40x magnification in classical pathology images, let's say, and then we can take the regions of interest as we like. I hope I answer the question. Yes, we can choose several regions of interest. This is very interesting.
Exactly. Some more questions coming in. What fluorophores can be used in the secondary antibodies?
This is important. So it has to match the emission and excitation spectrum of the filters that we have integrated in our instrument. So we optimized for
Dupouy, of course, Alexafluor 555, and Alexafluor 647. So as long as the fluorophore that is conjugated to the secondary antibodies covers that, then there is no real other specification for what kind of secondary antibody we need.
Great. Another question coming in. So how can one make use of this technology for cell line samples or cell suspension, as well as culture fixed cells?
Unfortunately, we don't have data on that, but we'd be happy to take this offline and come up with a project, for example, together to generate the data.
Does the repeating the elution process hamper the markers to be stained at the end?
Yeah, this is a very important point. So this is the reason why when we do the optimization for every single marker, we do it in a monoplex basis, but we do what we call elution efficiency and epitope stability studies. It will give us an idea of how far we can go in the elution for each epitope and allow us to decide whether we have to localize at the beginning, in the middle, of the end, or in the end of our eventual multiplexing experiment each marker. So yes, to some epitopes that can the elution can harm the binding. However, we can manage it by characterizing the marker during optimization.
Excellent. We are almost reaching the end. We have a couple more questions. So if you have any more, please send them now. The next question says, regarding the analysis of images, how many colocalized markers can be detected with your system? For most softwares, we are used to a maximum of 3 localized markers that can be detected.
Yeah, this is very interesting. I'm not sure I completely understand the question. So what we are doing is in every cycle, we get 3 images. We get one Dupouy image, marker A and marker B. We take the image, then we elute, and we stain again. So we are colocalizing at the same time, let's say, 2 markers plus Dupouy, but we do it over and over again, and we have our own proprietary stitching and alignment algorithm that will allow us to colocalize really all the cycles, therefore reaching the 40-plex.
And actually connected to this process, the last question asks, is the registration software included when multiplexing?
Yes, absolutely. So the software that we provide with the instrument will give the images already stitched, registered all together as one mosaic, let's say, in OME-TIFF format, so that you can downstream open it on any image analysis or tissue analysis tool that you'd like to use.
Great, this is very clear. So the Q&A is coming to an end. We have no more questions. So please remember that we will be sharing this recording on our company website, www.lunaphore.com. And if you would like to submit any more questions later on, please do so via email by sending a message to communications@lunaphore.com. Again, thank you to everyone for attending this webinar. Thank you, Dr. Dupouy, for this very insightful presentation and Q&A. And we're looking forward to receiving any additional questions from everyone else, and anything you might want to know about Lunaphore's technology, or if you're interested in possible collaborations, we are very open for discussion. So with this, I wish you all a great rest of the day, and thank you very much.